Well, it's a couple minutes after 11 o'clock Central, folks. I don't know what time it is where you are, but I do want to welcome everyone to the 16th Diatom Web Academy. The Diatom Web Academy has been hosted and put on by the Taxonomic Certification Committee of the Society for Freshwater Science and also diatoms.org. We've been meeting bi-weekly every other week during this pandemic time, because we want to make sure that we have a sense of community and communication amongst the diatomists all over the world. If you've missed any of these Diatom Web Academy presentations, we record each and every one of them, and they're available. You can find that at diatoms.org on the news page, or go to the YouTube, and we have a Diatom Web Academy channel that has all of our past presentations, and I'll have Jeffrey's here shortly. I'm going to turn things over to Sarah, and she's going to introduce our speaker for the day, and then we'll dive right in. If you would please enter your where you're checking in from over in the chat box, and if you do have a burning question during the presentation, feel free to enter it. Otherwise, we'll hold questions until the end of the presentation. Thanks, Sarah.
Thanks, Mark. And I'm delighted today to introduce Andy Alverson, who's an associate professor at the University of Arkansas. Andy works on the evolutionary history of diatoms, and he covers some just really fascinating and neat work. And today he's going to talk to us about the introduction and spread of a diatom in the US. And so thanks so much, Andy.
Thanks, Sarah. Okay, well, this is very fun to do. This is my first COVID talk on a video, so broadcasting live from my bedroom is very strange, but we'll give it a shot here. So this is a fun project for me to talk about, because this is sort of the first thing I discovered as an independent PI down here at the University of Arkansas. And so this has been kind of a project that's been evolving for several years, which I'll sort of recount in this talk. So I thought I'd start by raising an entirely non-controversial topic in the world of diatom evolution and biogeography. So the sort of dominant paradigm in this field was established back in the 1930s by Becking, who has this famous quote where he said that everything is everywhere but the environment selects. And he was a microbiologist, so he was referring here to microbes. And this has sort of evolved over the years into what is called the ubiquity hypothesis. And so this ubiquity hypothesis states that microbes, things that are less than a millimeter in size, is generally considered the cutoff, the explicitly stated cutoff, are in such high abundance that it makes for very easy passive dispersal. And this is also facilitated by the ability of many microbes to form resting cells. So you have this very high abundance, the ability to form resting cells, and so there are no barriers to dispersal. And if there are no barriers to dispersal, then microbes shouldn't show the kinds of biogeographic patterns that we're so used to seeing in things like plants and animals and in macroorganisms. So their distributions are constrained then only by the availability of suitable habitats. So there's also this idea of absence and proving absence for a microbe. So a proponent of this might say to someone who is claiming that a microbe isn't absent in some region. Well, maybe the habitat isn't suitable. Maybe it's there, but it's in low abundance. Maybe you weren't there at the right season. Is it there, but in a resting stage? And this is a wonderful hypothesis, because really the way it's been set up, you can't reject it. So you can really never satisfy the evidentiary demands for absence of a microbe. And I would point everyone toward a paper written by Dave Williams on this topic, which is a really insightful and thoughtful and also very entertaining take on this topic. So this, I would say, hasn't been, this idea, this paradigm hasn't been really warmly embraced by the diatom community. And that's because we see patterns that contradict this. So for example, we see taxa, species, genera, higher, that have restricted distributions. For example, like this Eunophora on the left that has been worked on extensively by Wim Viverman and others, that is found in New Zealand, Tasmania, and it's not found in the Northern Hemisphere. It seems to have a restricted distribution. And you see that this is a very easily identified, beautiful diatom. This is not the kind of thing you confuse with something else. There are also credible reports of endemism. And then there's also invasive species. So here this shows a picture of Thalassiocyra baltica, which Mark Edlund wrote about back in 2000, which is an invader into a recent invader into the Great Lakes here around Michigan in the United States. And this is, I think, a particularly compelling, in the existence of invasive species, I think, is a particularly compelling piece of evidence against this sort of everything is everywhere. And it provides really strong corroborating evidence for the natural patterns that we see with respect to restricted distributions and reports of endemism. And this is what I'm going to be talking about today. So this project began back in 2011 when I finished up my postdoc at Indiana University and moved down to the University of Arkansas. So for those who are outside of the United States, I am right here in the country. This is a beautiful area. This is right in the middle of the Ozark Mountains. And so when I moved here, I did what any self-respecting diatomist would do. I got in the car and drove around and started sampling the local habitats. And one of the places I went, so we're right here, I went up to Beaver Lake, which is a reservoir northeast of here that is dates back to the mid 1960s and is the result of a dam on the White River, famous for its sport fishing. And it's a really beautiful area. So I went there and took a plankton sample and came back to the lab and made slides using NAFRAX stolen from Ed Terrio, as all new, I think, investigators do. That's how we get our NAFRAX or HIRAX. And this is the sample I got. So you see that all of this other sort of stuff, sorry to show Nitzias so early in the morning, all of this other stuff just kind of fades away. And this big, prominent, beautiful diatom in the middle just jumps out. And this is clearly a thalassiosauroid, which is the group that I have worked on since I sort of started working in diatoms. And I had no idea what this is, which was kind of embarrassing. So I called up Matt Julius, a colleague up at St. Cloud State, and showed it to him, and Matt knew what it was. It's this species called Discostella asterocostata. And this is what I think would qualify in the literature as a so-called flagship species, because it's extremely prominent. All you have to do is look at this sample and it jumps out and it cannot be mistaken with anything else. So if anyone's worked and seen other species of Discostella, you know, we're not talking about fine scale differences between Steligera and Pseudosteligera and Walterecii. We're talking about a very prominent, unmistakable diatom, a so-called flagship species. These are some SEMs taken by Jeffrey Stone, and it shows the classic Discostella morphology. So it's got these strutted processes that are located in between two costae, which put it firmly in Discostella. And it's got long external extensions of those strutted processes that look like spines in LM. And it's got this strongly concentrically undulated valve face. So a really big, beautiful, prominent, unmistakable diatom. So that I knew absolutely nothing about. So I started digging into the literature on this and finding papers like this. So this is the paper where it was described originally as Cyclotella asterocostata from 2 reservoirs near Beijing in China. So this is exclusively, this article is exclusively in Chinese and Latin. And then there's papers like Cyclotella asterocostata, a little known steligeroid species from China by Hook, who is you know, one of the world's experts on Discostella. And then quotes like D. asterocostata has only been found in China, Korea, and Japan from Hook et al.'s Atlas of Freshwater Centric Diatom. So I was like, huh, well, that's weird. And so my very first student who worked with me was an undergraduate named Kylie Jones, and she just basically went around, drove around the region here and sampled all of the nearby reservoirs. And we found it in a bunch of these reservoirs in the surrounding area. And in 2013, she went to the North American Diatom Symposium in Maine. I couldn't go and I can't quite remember why. And she gave a poster at that meeting. And Jeffrey Stone, who probably many of you know and who gave a talk a few weeks ago in this series, visited her poster and was like, hey, I found this diatom as well in where he is in West Central Indiana. So that was interesting. And then I can't remember if it was at that meeting or later that we talked to another good friend, Kalina Manoilov, and she heard rumblings that this was turning up in some of the surveys, the USGS surveys around the Eastern United States. So the three of us compiled all the US records, which mainly come from collections that Jeffrey and I had made and from the NAQUA and Neotoma databases. And this is what the distribution of this diatom looks like in the US. So each one of these black diamonds is a record of a Discostella asterocostata. So far, we've mainly collected where I am and to the east. So a lot of my samples, Jeffrey's samples, and then most of these are from public databases. So it's pretty widespread in the Eastern United States. And this is not by any means exhaustive sampling. So from there then, we scoured the literature and found all of the records of this species, and as best as we could, tried to put latitudes and longitudes to those reports. And this map shows the global distribution. So all of the US records over here, and then all of the literature reports shown in black diamonds over here. So far, It's been found in China, Japan, South Korea, and there's one report from Eastern Russia. So a very peculiar distribution here, a really remarkable disjunction where we have it in the Eastern United States and out here in the Far East. So we were able to get the isotype for asterocostata. The type has apparently been lost, and it was described from 2 locations, and it's not really clear if it's a composite sample or not, but they were both around Beijing. So we got a sample from right here, and that's the only Asian sample that we've been able to look at. And when you look at this sample, when you just look at the slide under the light microscope, it looks like the flora of just any lake USA. It's got like all the stuff you're used to seeing. So I know this is very small, but it's got things like Asterianella formosa and Olicosyra ambigua, Cyclotella adamus, Cyclotella nana, Fragillaria crotonensis, you know, all the stuff we're sort of used to seeing in these kinds of samples, Nitsia palea, of course. So Kalina characterized the diatom communities from the sample from China and from a whole bunch of the samples that we've collected in the United States. And here's what the ordination of those sites looks like here on the left. So this is a non-metric multi-dimensional scaling of the diatom abundances from these samples. And the point is just to show that the sample from China is not some outlier in multi-dimensional space here, that it's actually very similar to a lot of the communities in the places where we've collected in the United States. So ecologically, it looks like the sites in the US and the one site that we have in China are very similar based on similarities in their diatom communities. And here's how the species fall out in ordination space. And I'll just point out right here, this DIAS, that's the star of our show here, Discostella asterocostata. And I'll come back to this briefly in a couple minutes. So we now see that there's this big disjunction, and we wanted to know, we had this hypothesis that it's probably a recent arrival in the United States. So the next natural question to ask is, if it is a recent arrival in the United States, like how recently did it come here? And so this is where the collaboration with Jeffrey Stone on this project has just been amazing. So Jeffrey did a bunch of coring around lakes in Indiana. And here is Jeffrey's student out on Beaver Lake, where I originally discovered it in Arkansas. So he cored some spots in Arkansas and around Indiana. And this is what those results look like. So these are the 8 different sites up here that Jeffrey cored. And here is the age of those cores. So these were dated. And the gray bar represents how far back in the core that Jeffrey looked, or how far back the core goes and how far back he looked for Discostella. And what we see is that in a few spots like Lake Fayetteville and Isaac Walton Lake, it's been increasing in abundance, and I'll show another site in a second where that has happened. But all things considered here, from the coring data that we have, which again is not exhaustive, but it points to, we don't find it any earlier than the mid-1980s. So it looks like an arrival as recently as the mid 1980s is what the paleo data say. So here's back to our ordination. There's this cluster of points over here on the far right of the first axis. And those are a set of samples from the core that Jeffrey took in this place called Dobbs Park Lake. And that is one of the places where this species is most abundant. And it's as you move up in the core, so going from further back in time to more recent, you can see that Discostella is increasing steadily in abundance and probably starting to edge out and outcompete some native stuff there. So cool. So what do we know? When did it show up in the US? So based on everything we know and with limited sampling, we know that most of the places where it's found are from impoundments and reservoirs or highly impacted rivers, that it appears to have shown up in the United States as recently as the mid 1980s. And there's one report in the literature that has a record in China that's dated back to 120 years. So some evidence that it was present over there before it was here. But again, this has all just been sort of side projects for us, so that we haven't had the funding to sort of do exhaustive sampling. But this is what we know so far. So the next question we wanted to ask, so we have this disjunction, and it looks like, according to the paleo data, that it's a recent arrival in the United States. So we wanted to ask whether we could detect any differences morphologically between the Asian population, the one that we have, and the populations in the United States. So this is what the LMs look like of a cell from the type locality and a cell from the United States. And people who have been doing this for a long time know that, yeah, those look really grossly similar, but maybe there might be subtle morphometric differences, which would suggest that maybe it's been here longer and that they've started to diverge. So to ask this question, we took a bunch of morphometric measurements of different radius measurements that mark discrete transition zones on the cell surface, and did counts of striae and strutted processes. And here is, so I'm going to show now some principal components analyses of those data. So anyone who's worked on diatom morphometrics or morphometrics of any organism basically knows that the first principal component axis basically always is measuring variation in size. So here we're plotting cell diameter on the x-axis and the first principal component axis on the y, and you see basically a perfect correlation. So 84% of the variation in our data is accounted for by PC1, and that is measuring size. So here is a plot of PC2 and PC3, so the next two axes, which account for a combined 9%. And the China, the individual specimens from China are shown in these diamonds, and individuals from, I think we have like 6 or 7 different sites in the US, not broken down by site, but just by country, are shown with these circles. And we see like no distinction at all between specimens from the US and China. So they're indistinguishable morphometrically. So what does this mean? What do these data mean? It, I think, is good corroborating evidence that they're very recently separated. Alternatively, you could argue that they've been separated for longer, but there's just ongoing gene flow that prevents them from diverging. But I think all of the evidence, when we look at it, points to a recent separation. So what do we know so far? So the geographic data point to this big disjunction: China, Korea, Japan, Eastern Russia, and the United States. I would argue that it's impossible that this has been overlooked in other areas. So places like Europe have, the diatom flora has been so extensively characterized, and this is such a distinct diatom morphologically that I think it's impossible that it's been overlooked. And I have queried other people in Europe, for example, and no one has been able to, no one has told me that it's been found there. The morphometric data point to a recent separation of the populations in China and the US, and that is supported by the paleo data, which point to a very recent appearance in the United States as well. And I should point out that the flora of the United States is very well characterized. So these are, the first records of this diatom in the US. So I think it's very unlikely, for example, that like Patrick and Reimer would miss this diatom. Okay. So how did it get to the United States? The answer is we don't know, but we can formulate a hypothesis. And so anyone who works in aquatic biology or just pays attention in the central United States knows that we have a big carp problem here. So we have 4 different species of invasive carp. They're collectively referred to as Asian carp. This silver carp shown here, this is, I think, this is some site in Kentucky, but if you drive a boat down the Mississippi River, in the wake of the boat, these silver carp just jump out into the water, and it just looks like, I don't know, something biblical in scope here. And so we're really afraid that these are going to make it up and into the Great Lakes, for example. So they're extremely high abundance. These things are everywhere. And so one question or one idea is that maybe these made it over here inside the guts of one of these carp when they were introduced. And there's actual literature that has looked at diatom survivorship inside the guts of specifically invasive Asian carp. And they showed, and we know this from lots of other studies, that diatoms can survive being ingested and then passed out of fish and aquatic insects and so on. So we know that this is a plausible vector. And so if we look at the distribution of these 4 carp in the United States, so we have 4 panels here. Each one of these panels shows a different species of invasive carp, and they're colored by watershed here. And so the distribution of the carp is shown in this filled in gray. And then our records of Discostella asterocostata are shown in these black diamonds. And you see for, all 4 of the Discostellas and for all 4, Discostella and all 4 carp species, there's pretty good overlap between the diatom and the carp. Again, the qualification is that we have not exhaustively sampled the diatom in the United States, whereas we have a very good handle on where the carp are. in the United States. But nevertheless, some pretty, I think, compelling patterns here. The other thing to remember is that we don't necessarily expect that the diatom will perfectly match the carp distribution, because once a diatom arrives here, it's of course free to disperse passively, irrespective of how the carp disperses. So nevertheless, like, you find places of co-occurrence of the carp and the diatom in the US. So in this next set of graphs, I'm going to show the global distributions of the carp and the diatom. And so the areas that are in this light green aqua color are where the carps, shows the carp's native range. And in this pink peach color shows where the carp is invasive. And I think this is a more sort of compelling set of plots. So here are those distributions for each of the 4 carp. And what we're really interested in here is whether the diatom co-occurs with that carp in its native range. And we see that for all 4 species, they do co-occur. So you see it for the big head carp. This black carp distribution is pretty compelling in its overlap. The grass carp and the silver carp also has a very compelling overlap in their distribution. So have we shown that it is a carp mediated dispersal? No, but I think this is a pretty good, at least hypothesis for how it may have been transferred here. So the mechanism of human mediated dispersal of this diatom. So that is the data that we have so far. We've been sort of writing this up as a COVID lock-in project. And so it's raised lots of questions of what we could do next. And so there are lots of good, I think, and interesting questions that we can address. So where did it come from and when exactly did it come? I think is a clear question. And I think it's one that we can very directly, and I hesitate to say easily, but it's certainly a question we could address very powerfully using population genomics. So we would, and under this kind of project, we would make samples from China, Korea, Japan, and the US. So it's native and introduced range. And this is very easily cultured, this diatom, which is very nice. It's easy to work with. And we could sequence cells from its native and introduced range. And we could also potentially isolate cells from the sediment records here and sequence those as well. And if we can't resurrect them, we may be able to just sequence the sediment from those regions. And there you have, if we can find the earliest records, that's when you have like a direct connection between the cells or population that was introduced and those in the native range. And so what we expect is that that there was a founder event here that resulted in a bottleneck. So in its native range, we expect to find a lot more genetic diversity than in its introduced range after it went through a bottleneck. And this is also, by the way, how we decisively test the hypothesis that this was an east to west dispersal, human-mediated dispersal, and not west to east. And it would also allow us to test whether there's been one introduction, or it's also possible, and say less parsimonious, but it's also possible that it was in the guts of potentially more than one different carp that was introduced here. So then we would like to know like where it's going and what it likes. So if we're correct that it showed up here in the mid 1980s or so, it's moved around pretty rapidly. And in some of the sites, we see it in pretty high abundance. So we expect that, you know, it's edging out native taxa potentially in those areas. And we don't know what the cascading ecological effects of that would be. But we could use, this is I think a good opportunity to use niche modeling and characterize the sites in Asia and the US to make predictions about where else it should be in the United States, and also and we could go and collect those and also make predictions about where we expect it might end up eventually. So niche modeling followed up with targeted sampling and coring. And so this is, you know, this is not really my major area of research, but as we've been writing up this paper over the past few months, it's got me pretty excited about the possibilities that this diatom and this event offer, and I think it could potentially be a very good model for understanding invasive species biology in diatoms and in general for microbes. The strengths of the system are it's an unmistakable diatom. I'm probably the worst taxonomist on this call, and even I can identify it. So it just can't be mistaken for anything else, which is a very good property. So we're not talking about different varieties of some Nitsia. We're talking about something that just can't be mistaken. The discreteness of the distribution is also, I think, a major advantage. So we have just these two discrete areas where it occurs, and it's not muddled by intermediate spots. So it rules out that there was some kind of stepping stone scenario. And so we have a very seemingly simple scenario to reconstruct by it being in two places and not 5 or 10. It appears to be a very recent introduction, which I think improves our chances of being able to reconstruct what the event was. We have a very strong hypothesis for the mode and direction of introduction. It's very easily cultured, so it has sort of weed-like properties, and it occurs in these disturbed areas, like where you find weeds. And it also has the advantage of having a pretty modestly sized genome. So the genome, we have a draft genome. A postdoc in my lab, Wade Roberts, has sequenced the genome of this, and it's about 100 megabases in size, roughly 3 times or 4 times larger than Cyclotella nana or Phaeodactylum. But nowadays, with what we can do with sequencing, this is, I would call this definitely tractable. I think there's a lot to learn generally about pretty basic questions in diatom biology and invasive species biology by this diatom that we're pretty excited about. Okay, so with that, I just want to acknowledge everyone who's worked on this. So a bunch of people at the University of Arkansas. Tyler Chafin did a lot of the GIS stuff. Kylie, my very first student, who did a lot of the morphometrics. Other people in my lab who have contributed in all ways to all projects. Jeffrey Stone's group at Indiana State University, the best thing that happened to this project was finding out that Jeffrey had found it and sort of combining our different areas of expertise. Kalina has been invaluable in characterizing the diatom communities. I could have never done that. And Matt and Ed and Kevin at Kentucky have also been valuable contributors. So with that, I'm happy to take any questions, and Jeffrey's also here I am not an expert in diatom paleo stuff, so he's here also. And yeah, thank you all for listening and happy to take any questions.
Thank you very much, Andy. If you guys have any questions, please drop them into the chat and I'll read them aloud. And while people formulate their questions, I just wanted to take this opportunity and ask, when you guys found the Discostella, it was found in river surveys and in these reservoirs. Was it mostly prevalent in the reservoirs?
I think that it's fair to say that most of the records are from reservoirs, and that is both here and in Asia. I think pretty much all of the records from China and Japan and Korea are from reservoirs.
Cool. And it's not really seen very often in Chinese lakes, or
so it's not like people have worked on this extensively, so we can't rule out that it's present in natural lakes. But one sort of frustrating part of this has been that we haven't found it in natural lakes. And so when you think about the paleo aspect of this, our records go back to the age of the system itself. So some of these reservoirs here go back to the 1960s, in some cases 1970s. So we don't have like deeper records that you could get from natural lakes.
Sarah's posted a wonderful screenshot of the diatom from one of her voucher floors. So maybe we have some more records of it. We have a question from David, and that is, any idea what is the closest relative and where does that live?
The closest relative of the diatom, so we don't know. We have put it into a phylogenetic tree, but the phylogenetic tree just has a few of the really common Discostellas in there, Stylidra, Pseudostylidra. So we can basically at this point say it's a Discostella. That is what we can say. We don't know what its closest relative is.
Alex says, what do the carp metagenomic data show, like from USGS environmental DNA sampling? Do positives for Asian carp match the diatom record?
Yeah, so they're using metagenomic data to see where the carp are, where we haven't actually potentially are, when in cases where we haven't actually seen the carp themselves. And so what I think is missing, Alex, is metagenomic data for the diatoms, where we could match up the distributions of the carp and the diatoms. So this is something I've thought about a lot. So there's been, and let me know, Alex, if I'm answering a question that you weren't asking. But
I guess can you hear me?
Yeah.
I guess what I'm saying is, in places like where is it, the Chicago Canal, and all the areas that connect the Mississippi to the Laurentian Great Lakes. I know that the USGS and the International Joint Commission, I mean, everyone's sampling like crazy. And could you obtain that data, for example, and use it as a predictor of where you might expect to find your Discostella species in that set, and then see whether it tracks up? And theoretically, it should.
I think theoretically it should. A couple things about that. I don't know if it's what they have is metagenomes, so actually sequencing the entire genomes, or if they're doing meta barcoding.
Yeah.
And if they're doing meta barcoding, then whatever primers they're using would just be like vertebrate or fish specific and wouldn't have the diatoms. But it's an easy enough
No. I'm saying get the locations, then go out there with your plankton net or your glue core or whatever, and then test them.
Sure. Yeah, definitely. That's totally possible to do. And, you know, I'm kind of surprised there hasn't been a large scale effort on this. If you look at like how much effort has gone in to sequencing the ocean and characterizing distributions of diatoms and other microbes that way, there's been no parallel effort to do that in fresh waters. So yeah, good question. And it's totally possible to do.
So while I'm on, Andy, I just want to say that you know, your presentation is fascinating and really clear, and I'm glad you touched on some of the molecular stuff, but it's also very and you said this in your introduction, these things embedded in sort of classic biogeographic theory pertaining to the microbial world. There's a bit of a connection. Many years ago, I worked with Deb Bhattacharya and Bob Anderson and Peter Siver and Wansoo Yoon on the Malamonas pseudocoronata species grade, which the morphology is a little bit less clear cut as in your case, and you know, it's a cyneurophyte, so it's not a diatom. But what we found is that neither the microbial biogeography nor Bos-Blekking appeared correct, in the sense that when we sampled, and we had Wansoo and his team of Korean phycologists, they sampled everywhere, and you know, Debashish, Indefatigable, Bob Anderson had his historical stuff, Peter Siver you just can't keep up with for the Eastern Seaboard. And what we found is that, you know, there was one phylotype, so we're talking about, you know, I think there were 5 or 6 genetic markers from the plastid, the nucleus, and the mitochondrion. And so it's pretty well supported molecular data set, again, largely from Wansoo's work at the Bigelow lab before he moved back to Korea. And there was, you know, the basal group, the basal phylotype was indeed cosmopolitan. But then there were also, within the sort of cryptic species complex of Malamonas pseudocoronata, there were definitely species, you know, genetic species or phylotypes that had biogeographies. And sometimes these arose in these ponds in Korea that were under intensive aquaculture, where water and fish were being exchanged, you know, between them, and yet you'd have 2 distinct phylotypes, and you'd have a third pond that would have the same phylotype as Northern Europe or Florida. And the only way we could, and so basically the conclusion was that neither Bos-Blekking nor the strict biogeography, or, you know, microbes have biogeographies, which is like what you show, which is what like every hot spring diatom assemblage shows, that neither is necessarily correct. And the only way I can look at it in my simplistic geological perspective is that this would appear to mandate that these phylotypes have evolved on evolutionary or tectonic timescale. So we're talking, you know, tens to hundreds of millions of years, and not a matter of, you know, decades to centuries, which I guess is consistent with, you know, you don't get a lot of sex when you're cyneurophyte. So I just thought I'd throw that in, and if you want to continue that, but it's you know, it's not from the diatom world, but it's still from the chromist world.
Well, yeah, it's from the microbe world, and I mean I think that it shouldn't have to be a choice between the 2 extremes. You know, the interesting thing about these samples, you know, we have this one sample from China and these other ones, is that observation that the sample from China is full of taxa that are, you know, putatively like cosmopolitan and sort of fit the Bos-Blekking paradigm. So you see, like, you see all the stuff you would see over here. And then like the outlier in there is this Discostella, which, you know, I've sampled a lot of lakes and stuff around the US, which you don't see. So it is this, like, these samples are really cool because they are sort of this really interesting blend of cosmopolitanism and then what appears to be this really discrete human-mediated break in that pattern. So yeah, I don't think it has to be one extreme or another. The other thing I feel like with work by Ed Terrio, and then what Jeffrey talked about a few weeks ago on this in this forum, is that parapatric speciation might be pretty common in diatoms, which it sounds like maybe what you were seeing in Malamonis as well.
Yeah. Thanks, Andy.
Yeah, cool.
Great.
Thank you.
Wonderful discussion. I have another question here from Mark, and Mark asks, has the invasion followed a typical boom and bust invasion scenario anywhere?
Jeffrey? I don't know that we have the data for that.
Okay. And if Jeffrey's replying, he's muted. Oh, there he goes.
Yeah.
The Fayetteville Lake sample has a really, it seems like the asterocostata just has taken over that system, and in a really short period, maybe a decade or something. The rest of the records are a little less clear.
So it's still booming or just starting to boom.
So it's starting to boom, and maybe we haven't seen the bust phase of that yet.
Sarah just wanted to let you know, great talk, and the voucher flora images that she posted are from lakes in the northeastern United States, and she will follow up with localities for you later.
Yes, thank you. Fantastic. Yeah.
I'm going to go straight to my tattoo artist.
Yeah, exactly. Me too. And Anna, yes, that was my cat. My cat sleeps for 23 and a half hours a day and then knows when I'm on a Zoom.
Andy, I wanted to ask you a quick question for the collection of morphological data. What process did you guys use, and were there any bits of software or any kind of automation that you might encourage other people to use if they're trying to quantify populations of diatoms like this?
So we used ImageJ, which is a freely available software distributed by the NIH here, so anyone can use it. And with that, it was very easy to say count all those things in a very easy way and to measure those diameters. The other thing that's really nice about doing those analyses on digital images is we have all the digital images. So I was able to go back, for example, as we were putting all of this together and see that an undergraduate had included like a Steligera type thing in there. And so it was very easy to reconstruct and reproduce everything. So those digital images are great for archival purposes, and the ImageJ was really helpful. Jeffrey used some, I think, Leica software that is made by Leica for your morphometrics. Yep.
Is he shaking his head? I can't see him.
I think he's shaking his head, yes.
Okay. He could be nodding to the music. I don't know. One more question here from Mark Hedlund. Invasion conjures up words like mean and nasty. Any thoughts on whether Discostella asterocostata is having any negative ecological impacts or the mechanism of impact?
So I think we don't know, but these systems are probably zero sum with respect to the diatom numbers and communities that they support. So as Discostella is going up, is booming, say in Lake Fayetteville and some of the other places that Jeffrey has surveyed, concomitantly, other things are going down. So what the ecological consequences of this are, I don't know. but it certainly could have an impact on the biodiversity. You know, another thing that's interesting to think about here is that, you know, we've started to have a better appreciation for all of the stuff that is attached to diatoms. So we, you know, we think about the carp as a potential vector for the diatom, but the diatom was potentially a vector for other things. Could have had a chytrid attached to it. It certainly had a whole bunch of bacteria attached to it. And those are even less, less visible and less easy to characterize, but it's potentially as or more important than what we've seen with the diatoms. Any alternative explanations? Any alternative explanations? At the moment, no. But I would love to hear if anyone has ideas on alternative explanations. I think it's very, very unlikely that it's always been here and has always been overlooked. I think that is extremely unlikely, but as a mechanism for how it got here, I don't have any other ideas, but I would love to hear if someone has any ideas.
Another question from Alex is, are there any reports from South America? And I'll say
not published as far as I'm aware. It's possible that I could have missed something. I'm definitely open to that possibility, but insofar as I'm aware, no.
I'm just, yeah, I just like, you know, I've seen something like this before. And when I saw your very first slide, I said, you know, I was thinking like, oh, wow, that, you know, looks distantly like Ellerbeckia arenaria formateris or something from my childhood.
Mm-hmm.
But, you know, I have, and we did have these lakes, really high up lakes in the high Andes, in the Altiplanos. I don't know, maybe check with somebody who would know, like Sherry or something like that, because she would have seen it. But
okay,
yeah, I've seen it somewhere. I just can't place it right now.
Okay, yeah, cool. Yeah, that'd be great.
We'll need confirmation because, yeah, you're wrong. I'm a worse taxonomist than you are.
Yeah.
We have two more quick questions here, and then we'll wrap things up. I got one question asked to me privately, and that is, does this diatom have a particular microbial population associated with it? Jean Marie asked that.
Don't know. Hasn't been characterized by us or by anyone else that I'm aware of. So don't know the answer to that, but it's a very good question.
And then Anna would love if you could maybe drop in the chat really quick the reference for the everything is everywhere idea.
I'm gonna, Anna, there's my email, and if you email me, I'll send you stuff.
All right. Well, thank you again, Andy. This is a wonderful discussion.
Yeah, that was fun. Thank you, everyone.
And just later today, we'll have this recording uploaded, and I'll link you guys to the YouTube playlist again. And if you don't want to memorize that URL, you can always go to diatoms.org and find all of our previous talks and what is upcoming. Thank you very much, Andy.
All right. Thanks, everybody. Take care.